How's it going, guys? All right, we're going to take a look at chapter 13, which is not one of my favorite chapters. This is actually, I think, a very complicated chapter that I won't, and we'll have a couple of the questions, but these are not some of my favorite questions that might pop up on our written part of our test. But we'll pull a few from the online test too, and I'll try to find a few that work. I'm going to share the screen with y'all. So if y'all would, hang with me. We're going to go to chapter 13. Okay, y'all should be able to see that now. Okay, all right. So this is kind of, remember the big one in chapter 12 was protein synthesis, right? And that's going to be a huge question on the written part of the test and quite a bit of the multiple choice. But in this chapter, this is looking at how we can regulate genes. And I think it's pretty tricky chapter because we don't know how this all works. So a lot of this is going to show us some clues as to how this might go down. Okay, and get this thing to work. Here we go. All right. So we're going to look at prokaryote regulation, which are simple. When those are our bacteria, they're a little simpler than we are. And we're going to see that some of the first research discoveries of how genes are expressed was found in that. And we'll take a look at a couple examples of that. Then we'll look at what we know about eukaryotic regulation, and then we'll talk about gene mutations. One of the more interesting commentaries in the book is this first one on the first page on page 228. It talks about social status can affect gene expression in female monkeys that have a dominant hierarchy compared with males in the wild. In this experiment, they showed that higher status females, monkeys had stronger immune systems and genes that were expressed differently, meaning different genes were turned on and off than lower status female monkeys. This shows that many factors, including social status and even can control gene expression. My daughter is taking a class at Ole Miss, and they're talking about how living in a poor environment, rough, worried about food, very poor, makes them have more genes turned on that make them sicker. So how our genetics can be involved by our status. Okay, so with all this craziness of our two-party system and this election coming up and COVID, this can make us all possibly be more sick, and that's kind of scary to think about. Okay, all right, but looking at prokaryote, the big one here, the first question on our review sheet too, describe the operand model of gene expression in prokaryotic organisms, and this is actually Jacobs and Monin, I believe, proposed this in 1961, that gene regulation on a single on a bacterium, which has one chromosome, right? And we know that certain enzymes are required for this to happen. They proposed that there were several players involved, and so the operand is a group of structural and regulatory genes that function as a single unit. Let's take a look at one. They may involve pieces on the DNA called a promoter sequence, a operator sequence, and then the actual structural genes themselves. And then somewhere else on the chromosome will be a regulatory gene that can code for a repressor protein that can turn on or off these structural genes. So basically what it's saying here is in an operand, you got all these players involved, the promoter, the operator, the structural genes themselves that we want to transcribe and translate, and regulatory genes. And they kind of draw it out. We'll just take a look at it in the first one that they discovered here, called the TRIP operand. Okay, so this is a regulatory code for a repressor that if tryptophan amino acid is absent, the repressor is unable to attach to the operator. Okay, and RNA polymerase binds the promoter, and enzymes needed to make tryptophan are produced. If tryptophan is present, it binds for the repressor protein that sticks to the co-repressor, and that cuts off and prevents the enzymes needed to make tryptophan. So the repressor is going to block the synthesis of the enzymes in the pathway to make tryptophan. Okay, let's take a look at it. So here is the regulatory gene making the active repressor. Okay, and when it binds here, it prevents transcription. So if you have plenty of tryptophan in your area, in your cell, then you're not going to make it. Okay. Now take a look at here. In the absence of tryptophan, the enzyme, this falls off, and then the enzymes needed to make, these are the enzymes needed to make tryptophan occur. But when tryptophan is present, tryptophan binds to the repressor, sticks it, makes it bind in, and then cuts off, preventing RNA polymerase from coming in and to the promoter and transcribing. And we got transcription and then later translation of the enzyme. So that's showing the messenger RNA being made, and then in translation would build the protein. So this is a cool one, right? So in other words, in normal situation when they have plenty of tryptophan in their diet, here's what's going on, right? Plenty of tryptophan is present, shuts down the system. No tryptophan, they need to make some, then the enzyme, without tryptophan being bound here, it falls off the operator and the promoter comes in, RNA polymerase finds the promoter and the operator, and we make the genes, these or enzymes needed to make tryptophan. So then the other one that the more famous one I'd heard about was the lac operin. Okay, and this is one of the first ones because this one has the regulator codes for a repressor. So if lactose, a sugar that can be used for food, is absent, the repressor attached to the operator and the enzymes to break down lactose is not being made. If lactose is present, it combines with the repressor and renders it unable to bind to the operator. Thereby, RNA polymerase comes in and works. Okay, take a look. This works a little bit opposite. Okay, so you can see here that that as long as there's plenty of food to eat, there's no problem here, right? This thing just keeps rolling. There's nothing being made. But if you have lactose present, then lactose will bind, pull that off, and then we make the enzymes needed to break down lactose. Okay. They further discovered that this lactoperin in E. coli preferentially break down glucose. So lactoperin is maximally activated only in the absence of glucose. When glucose is absent, cyclic AMP, which is a real famous second messenger, this is probably the one of our 24 or 5 hormones that we have in our body. Probably half of them bind to receptors and trigger the production of cyclic AMP. So it's real famous. It's called the second messenger because it's the continuation of the hormone inside the cell. Well, here it is in bacteria. It is being produced, and when it binds to this cavalite activator protein, CAP, the CAP then, when bound, binds to a site near the lacto operator, and this makes the RNA polymerase come in and bind the promoter, so it enhances the production of the enzyme needed to break down lactose. When glucose is present, there's little cyclic AMP, the CAP is inactive, and the lactoperin is not expressed maximally, although it could be being expressed a little bit being made. So here we go, inactivated, and they're going to show it here. Here we got in the presence of lots of cyclic AMP, the CAP binds and it activates, and this is going to enhance the lactoperin to turn on the production of the enzymes to break down lactose. Okay, so lactose presence, bam, more activity. Lactose present, glucose present, cyclic AMP is low. So if glucose is present, then it's not going to bend, and we're not going to get this coming in as well. Kind of wild. I know, very complicated. Like I said, I'm not going to go crazy on that, but just deal a little bit about that. Okay, and then if we look at this next thing, it's talking about, now we're looking at, and then we get the part, and we're talking about eukaryotic regulation. It talks about these different levels of control. So think about this. We'll look at a picture that kind of shows it actually a little better than this. But obviously, we could have nuclear level where the chromatin structure could prevent expression of a gene. You can have, that's the folding here. Then you can have something that stops transcription. Then you can have something that screws up the introns being cut out, right? Remember that post-transcriptional control. And then something could just keep the messenger RNA from going out there, right? And so translation, something stops translation. And then after the protein's made, we can have post-translational control where the proteins are not folded properly, and are they not carried out of the cell by their Golgi apparatus, et cetera. So we have, folding of that 1, 2, 3, 4, 5, 6 levels, at least 5 or 6 levels of places we could have something prevent the transcription and translation of the protein. Okay, the first one they look at here is chromatin structure, and I think this is pretty cool because if you remember, chromatin is our DNA when it's unraveled in the nucleus. Okay, and then it folds up on these histones and forms these nucleosomes. Okay, so the level of chromatin packing can determine the degree of nucleosome coiling. So in other words, take a look. So these highly coiled ones here are not going to be opened up and allowed to be transcribed, but if they're open, then they can be transcribed. So the way that the protein, the DNA is folded up on the spools of histone can regulate gene expression. Matter of fact, this euchromatic is loosely coiled DNA that we know is transcriptionally active, and heterochromatin is tightly packed DNA that is transcriptionally inactive. And you can see that in the cell. Here is the heterochrome, right? And it is not going to be just so packly tight. And this other is loosely tight, the euchromatin, and it then is transcribed. A good example of this is in the females. Y'all have 2 X chromosomes, but you actually only need 1 to be active. So when you start making body parts and stuff, one X chromosome is tightly packed up and chunked over the side in the cell and forms what's called a bar body. And so you only use 1 of your X's. Now men, we have to use the one we got, so all of our cells express that. But in girls, half your body expresses 1 X from mama, and the other half of your body expresses the other X you got from your daddy. And a great example of this is the calico cat color, which has a patch of orange and black. In the males, it will be one color, but in the girls, you get, because of the two chromosomes, you get a hodgepodge here of that. Take a look. So the one on the top, the orange, has the active, active one of them, the one that has the orange gene is expressed, and then the one on the bottom there, the black gene is expressed. Is that not cool? So this would be her DNA, and half her body active uses this one, half uses this, and voila, you get the beautiful coloration that we see in the calico cats. The males will be one color. Pretty cool example. Okay, now in your book, this is another cool commentary. Like I said, this is a tough chapter, but I was really enjoying reading this. I was going to show you this, called epigenetic inheritance. This blew my mind. This is on page 236. But here, let me define it to you here, and I'm going to read it to you. I'm going to read the definition out of the book here. Epigenetics are heritable changes in gene expression without changing the DNA sequence. Chemical reactions due to environmental exposure influence how genes are turned on and off. They are weakened or strengthened, and how they change our immune system and how they build muscles and brains and other body parts. To look at this, they looked at identical twins here, and they studied, assuming they were treated equally by the parents. They saw over time that certain parts of their genes are what we call tagged, and this tagging is where a methyl group is attached to the cysteine in the DNA. And with a methyl group attached, transcription cannot occur. This methyl group interferes with transcription factors and other proteins in transcriptional machinery, thereby silencing or weakening a gene. Over time, the differences in these tags accumulate, making twins increasingly different from each other, as shown in the pictures here in our book. And they actually show on the picture, look on page 236, and it has comparisons of the twins' chromosomes. And the 50-year-old twins have more epigenetic tags in different places than do the 3-year-olds. I thought that was pretty fascinating. So your environment does have an effect, okay, and on that, and so this is good, because then theoretically then we may be able to make some drugs. For example, read this last part here. A study using rats showed that rat pups that are licked and nurtured by their parents become calm adults. Rat pups that are not nurtured or have anxiety or anxious. Injecting a calm rat with a drug that adds methyl groups creates an anxious rat, not a cat, Conversely, injecting an anxious and anxiety and anxietized a worried rat, okay, with a different drug that removes the methyl group creates a calm rat. Let me have treatment for our anxiety here, people. Okay, wow, that's on page 236. Check that out. But I think this is cool. So it explains how unusual inheritance patterns that we see may also play a role in growth, aging, and cancer. So how we can be even identical twins. Environment can methylate our cysteines in our DNA and cause genes to be not transcribed and translated. Woo. Okay, transcription control, that's pretty cool, right? Makes sense where something prevents the transcription from occurring, okay? And when there are all these transcriptional factors that we have to have, so if we don't have these factors, then transcription can't occur. That's known to be part of it. And then post-transcriptional could be not cutting those introns out, right? And that's a big deal. Remember, those introns have to be removed. Look at that. The hypothalamus gland and the thyroid gland produce a protein hormone called calcitonin, but the messenger RNA that leaves the nucleus is not the same in both types of cells. The thyroid and hypothalamus therefore release different versions of the hormone. Wow. So pretty cool. So all about how the splicing occurs can alter the genes, okay, and that can include making alternate forms of a product, as we see here. Okay. Now another really exciting area of this is on page 237. It talks about the small RNA molecules. Okay. Now I want to read this. I don't always read y'all, but I thought this is pretty exciting. Let me read this to you. For a long time, scientists were faced with a mystery. A cell appears to contain vastly more DNA than we need to account for the number of expressed proteins. The DNA that was not expressed was initially called junk. But recent scientists have begun to understand the role of this DNA. Although only 1.5% of the transcribed DNA code for proteins, the remainder is used to form small RNA molecules. We now know that these small RNA molecules represent an important form of gene regulation that functions at multiple levels of gene expressions. Okay, so take a look at some of that, and then he talks about this. So some of these things, these are sources. They can regulate translation by causing destruction of messenger RNA. They can cause alternate slicing. Okay, and matter of fact, if you look in your book on page 238, it talks about this RNA interference. By using a combination of these messenger RNAs and these small interfering RNAs, a cell can fine-tune the amount of product being expressed from a gene, much as a dimmer switch on a light regulates the brightness of the room. both this microRNA and the small interfering RNA molecules. That's both these. Okay, say it again. Most of these microRNA and small interfering RNA molecules interfere with the normal gene expression pathway, and this process is referred to as RNA interference. It mentions here the first scientists to artificially construct these miRNAs and siRNA molecules to suppress the expression of a specific gene were Dr. Fire and Mello, and they won the Nobel Prize for Physiology and Medicine in 2006 for this discovery of a drug to suppress the expression of a disease-causing gene. So fascinating area. So what we're saying now is a lot of this DNA that we didn't know what it did, it's all about regulation, because we have not figured out how regulation occurs. And so when I start reading out of the book, this is new stuff to me. So this is the cutting edge of what's going on here. And it looks like a lot of gene expression is controlled by these sRNAs, okay? Small RNA molecules, specifically these microRNAs and small interfering RNAs. Wow, fascinating. A lot more to be discovered about that one, but I just want to mention that to you. Pretty wild. And it shows how some of these work. Kind of shows how that can stop the, look at that, alters the DNA there. It can, and here it can bind that, prevent translation, or just have the messenger RNA broken down. That's probably what a lot of that DNA is doing that we don't know about, that we don't know it does not transcribe into a particular protein. Okay, here's another one showing alternate processing. So we can make different proteins from the same gene by splicing the RNA differently, cutting out different introns. Pretty wild, okay? Then of course, translation control, something that stops that. And talking about this is pretty famous about these, the presence of this 5' caps, got to be there for that to happen. Post-translational control, proteases break down the proteins, and they have to be, these are enzymes that break down proteins. So maybe the protein's made and then the cell just destroys it. So that would be a post-translational control. And here's showing that protease breaking down the protein being made here, and here it is destroyed. So that's an example of that. There's still more to be discovered about that, but proteases breaking down proteins seem to be pretty wild. Okay, and then the last little part just talks about a gene mutation is a permanent change in the sequence of DNA. And sometimes, because remember, we have some of the amino acids have the same codon, so no effect on protein activity can occur. But sometimes you can have complete inactivation of protein, or you could have some new structure formed, okay? So germline mutations occur in sex cells, somatic mutations occur in the body cells. Okay, now we can have spontaneous mutations that occur about 1 out of 1 billion nucleotide pairings, just A binds to C instead of T, okay? But induced mutations are caused by mutagens such as radiation, organic chemicals. Many mutagens are carcinogen and are cancer causing. Some environmental mutagens include ultraviolet radiation, tobacco smoke. Also, I was reading in here a chemical that they put in the gasoline that we all pump, right? And it's also a known carcinogen, okay? Yeah, this is pretty scary, okay? And so this is why there's that little warning label, if you'll see it outside, ethylene dibromine, which is added to leaded gasoline to vaporize lead deposits in the engine and send them out the exhaust, is a dangerous one. And some fungicides mentioned here and some drugs for TB are known to be mutagens, okay? So an induced mutation is one caused by something, experimental exposure. One of the ways we can see if a chemical is a mutagen is this Ames test, okay, and it shows it here, where suspected chemical is added to this strand of bacterium that require histamine. The plate then onto, then you take the control bacterium strain requiring histamine, both, but here you put the chemical in, and then you look for mutations. And mutations here on a plate that lack histones means they start growing, they've mutated and started breaking down, they made histine so they can then live. So that tells you that chemical is a mutagen if you get growth on that plate. That's called the Ames test. Now that's pretty cool. Okay. A point mutation would be one nucleotide replaced, okay? A frame shift would be the insertion or deletion of a nucleotide. Now look what happens. If you get a point mutation, then it would only affect one amino acid, or it may not affect any if it's a codon that still works. And remember, because some of them, like some of the amino acids have 4 or 5 of the 64 codons just code for them. So you can have, that's called a silent mutation, when one of those happens and nothing happens, okay? But if it does, it would just screw up that one amino acid, right? And sometimes we'll see in a minute that can be really bad. Frame shift mutations are worse because then you actually change up all the spelling. For example, they put this in several books. The cat ate the rat. If you delete what is that? The C in cat is the ada ta ha ah. Okay. Or if you insert a C for the same place, now it's the CCA tat atha urate. Completely spell everything because every 3 letters is a different code and it's completely altered. So may you never get a frame shift mutation. If you do, that's really bad. But here's an example of a point mutation. The normal hemoglobin, which I think is about 120 amino acids long. Normal hemoglobin has protein, glycine. Glycine here, valine substitutes for glycine, and you get this one error here, and that causes this terrible disease where the red blood cells are not. And you can show it here. They're going to show on how this happened, but that's the red blood point mutation there in hemoglobin, causing that terrible disease. Okay, and then the last little section is just talking about other ones here. Mutations can cause cancer. Okay, proto-oncogenes are cells that can control, or genes that control cell division. If they mutate, then they become cancer. And in tumor suppressive genes, if these genes go out of whack, then you can lead to tumors into cancer. Nothing much there. And just talks about just non-functional proteins. Not my favorite chapter, but hey, this chapter is growing. It went in the book when I took this class a long time ago. All right, that's all I got on chapter 13. Not my favorite. Won't see a lot of them on the test, but at least we went through it and had a little something on it. All right, y'all have a great day, guys. I'm going to stop this thing. I'll go try to work on 14 and 15 for next week.